2. The genetic code 3. Components required for translation 4. Codon recognition by tRNA 5. Steps in translation 6. Antibiotics that target translation 7. Post-translational modifications. 1. Overview. Genetic information is expressed/passed through transcription to RNA and, in the case of mRNA, subsequent translation into proteins. The process of translation requires a genetic code, through which the information contained in the nucleic acid sequence, mRNA, is expressed to produce a specific sequence of amino acids. 2. The genetic code. The genetic code is a dictionary that identifies the correspondence between a sequence of nucleotide bases and a sequence of amino acids. Each individual word in the code is composed of 3 nucleotide bases. These genetic triplet sequences are called codons. A codons. 61 of the 64 codons code for the 20 common amino acids termination, stop or nonsense, codons. 3 of the codons, UAG, UGA, and UAA, do not code for amino acids, but rather are termination codons. When one of these nonsense codons appears in an mRNA sequence, it signals that the synthesis of the protein coded for by that mRNA is complete. B. Characteristics of the genetic code. 1. Specificity. 2. Universality. 3. Degeneracy. 4. Non-overlapping and anomalous. C. Mutations. A. Change in DNA sequence. 1. Silent mutations. Frameshift mutation. 3 components required for translation. A. Amino acids. All the amino acids that eventually appear in the finished protein must be present at the time of protein synthesis. A note: if one amino acid is missing, for example, if the diet does not contain an essential amino acid, translation stops at the codon specifying that amino acid. This demonstrates the importance of having all the essential amino acids in sufficient quantities in the diet to ensure continued protein synthesis. B. Transfer RNA. At least one specific type of tRNA is required per amino acid. In humans, there are at least 50 species of tRNA, whereas bacteria contain 30 to 40 species. Because there are only 20 different amino acids commonly carried by tRNA, some amino acids have more than one specific tRNA molecule. This is particularly true of those amino acids that are coded for by several codons. 1. Amino acid attachment site. Each tRNA molecule has an C. aminosyl tRNA synthetases. This family of enzymes is required for the attachment of amino acids to their corresponding tRNA. Each member of this family recognizes a specific amino acid and the tRNA that corresponds to that amino acid, ACE-accepting tRNA. D. Messenger RNA. The specific mRNA required as a template for the synthesis of the desired polypeptide chain must be present. E. Functionally competent ribosomes. 1. Ribosomal RNA. 2. Ribosomal proteins 3A, P, and E sites on the ribosomes. 4. Subcellular location of ribosomes F protein factors. GATP and GTP are required as sources of energy. Cleavage of 4 high-energy bonds is required for the addition of 1 amino acid to the growing polypeptide chain, 2 from ATP in the aminosyl tRNA synthetase reaction, 1 in the removal of PPI, and 1 in the subsequent hydrolysis of the PPI to inorganic phosphate by pyrophosphatase, and 2 from GTP, 1 for binding the aminosyl tRNA to the A site and 1 for the translocation step. See figure. A note: Additional ATP and GTP molecules are required for initiation in eukaryotes, and an additional GTP molecule is required for termination. A. Anti-parallel. Non-traditional base pairing with the 3 base of the codon, the last base of the codon. This movement is called. The mRNA is translated from its 5N to its 3N, producing a protein synthesized from its amino terminal N to its A. Initiation of protein synthesis involves the assembly of the first codon in the message, GTP, which provides energy for the process, and initiation factors that facilitate the assembly of this initiation complex. In eukaryotes, the initiating AUG is recognized by a special initiator tRNA. Recognition is facilitated by EIFs. Site and GTP is hydrolyzed to GDP. After the peptide bond has been formed, the ribosome advances 3 nucleotides toward the 3 end of the mRNA. This process is known as translocation and requires the participation of EF2 and GTP hydrolysis. This causes movement of the uncharged tRNA into the ribosomal E site before being released, and movement of the peptidyl tRNA into the P site. C. Termination. Termination occurs when one of the 3 termination/stop codons moves into the A site. 6. Antibiotics that target translation. The initiation process in protein synthesis is different in prokaryotes and eukaryotes, as shown in the next slide. Many antibiotics that are used to combat bacterial infections in humans take advantage of the differences between the mechanisms for protein synthesis in prokaryotes and eukaryotes. 7. Post-translational modifications. Post-tRNA modifications. A. Trimming. B. Covalent modifications. Proteins, both enzymatic and structural, may be activated or inactivated by the covalent attachment of a variety of chemical groups. 1. Phosphorylation. 3. Hydroxylation. 4. Other covalent modifications.